So I have the privilege today to introduce the first plenary speaker. And the first plenary speaker is Bess. And while preparing, thinking of what could I tell about Bess, I was looking back and I found out that I met her for the first time 27 years ago when I came to Scripps as a postdoc working in Farouk's lab, and she was there as an associate or assistant research oceanographer in the food chain research group where Dick Apley and a lot of other heroes in oceanography, they were sitting door to door, and I was really astonished getting to know all the people at once who were familiar from textbooks. And Bess was back then working on nitrogen using immunofluorescence to quantify nitrifying bacteria in the Pacific. And she's still doing this. She's still using other methods, but she's still working on the nitrogen cycle. And over all these years, the nitrogen cycle has been received major alterations. The view on the nitrogen cycle has been altered, and a lot of the revolutionized view we have on the nitrogen cycle in the marine environment really comes from the work of Bess. Bess received her PhD thesis or the PhD degree in biological oceanography at the University of Seattle, and thereafter she joined the Scripps Institution of Oceanography, and after a few years in 1989 she moved to the University of California, Santa Cruz, so just a bit further up from San Diego to Santa Cruz, starting up as an assistant professor, but leaving then essentially Santa Cruz, the University of California at Santa Cruz, as a professor to join the Princeton University, where she is a professor now in the Department of Geosciences. Bess Ward's scientific work has been acknowledged by numerous awards, among others the G. Hutchinson Award, which she received in '97. And I think today she will talk us and introduce us to the nitrogen cycle and the way we look at the nitrogen cycle at the moment, and there is no better person to tell the story than Bess. Please welcome Bess.
Okay. Right. Well, I'd like to thank the organizers of the whole meeting, because it's really been a nice one, a great meeting and a great place. And I know it's a lot of work for them, and we take that for granted. So I would like first of all to thank them, which I'm very grateful for that. When they asked me to give this talk, they gave me a title that they would like for me to speak about, and it was not that one. Novel microbial metabolisms and interactions in aquatic systems. And that didn't really sound like me, but as Gerard told you, I decided to talk about the nitrogen cycle because that's the only thing I know anything at all about. But there's lots of novelty and lots of interactions in the nitrogen cycle. And as he also said, especially it seems in the last few years, but I still chose the title Cryptic because I am often struck by two things. Although we've been studying the nitrogen cycle for a long time, and he almost told you how long I've been doing it, we still don't know some pretty obvious things. So cryptic meaning that there's a lot hidden behind some really obvious patterns. And I'm going to talk about two examples that I have been working on recently and hope to continue to work on in the future. And these are they. I understand that the pointer doesn't work, so I'm just going to gesture my arms and try to tell you what to look at when there's something important. So the two systems I'm going to talk about are nitrogen utilization patterns in the surface ocean. This is largely the work of my former student, current postdoc, Sarah Fawcett, and our work with our colleague Danny Sigmund at Princeton. And the second one is nitrous oxide fluxes in the OMZs, which is the current work of several people in my lab. And you'll see the names of everybody who did the work on the slides where their data appear. So the reason to work on nitrogen in the first place is that it is one of the most important limiting nutrients in the world ocean. No matter how you look at it and how many ways you reanalyze the data, nitrogen keeps coming up. It's especially recognized as important in everybody's favorite piece of ocean, the North Atlantic, as you can just see by the number of dots where you're able to do this kind of analysis. And so I'm going to start with another favorite, already mentioned by Matt, as the BATS, the time station in Bermuda, as one of the places that we see some amazing and unexplained stuff. So Matt already mentioned the value of time series, and I just want to say that I agree with that. I only have one complaint about this figure, which I lifted directly from Mike's paper, from that really nice recent review on the BATS time series. They published salinity, temperature, and density figures like this in color, but not nitrate. What were they thinking? You know? So nitrate, to me, as the biologist, really defines the system, although, of course, the physics probably determine the nitrate. But what I want you to see here is that nitrate is, there's usually not very much nitrate at BATS in the surface layer. Every now and then there's a seasonal or episodic intrusion, and you get maybe up to 50 nanomolar. But usually it's very, very low, and so the nitrate is basically far removed from the surface layer, where the light is, where the phytoplankton need it. Nonetheless, you do see a seasonal cycle, and Matt showed you a really nice one from the hot station. I just have two comparison points for this one. A picture from Fred Lipschultz's summary work. And you see that in March, when that nitrate intrusion often happens, you have increased primary productivity, and that would be on your left. And most of that is supported by nitrate assimilation in the dark squares. Whereas in September, when it's been highly stratified for a long time and there's no nitrate left, if there ever was any, the primary production rate is much lower, and it's supported mostly by ammonium assimilation. So you do see a seasonal signal, but you usually see almost no nitrate in the system. And this is another way of looking at that. These are data presented by Sarah Fawcett. And it's just a depth profile as an example of what you would also see in that contour map. And you never, it's almost safe to say that you never see nitrate in the surface water. But there is evidence in the nitrate isotopes that nitrate has been there, or that there is at times that 50 nanomolar or so, and it comes in the evidence of the natural abundant stable isotopes of nitrate. And the deep water value is about 5, just about everywhere. Up in the thermocline of the BATS region, it's about 2.5. And when you get into the surface layer, if there's enough nitrate to measure it, which Sarah and Danny can do with the denitrifier method on a really small amount of nitrate, you see that it's often highly enriched, and that's evidence that phytoplankton have taken it up, and the fractionation effect is being expressed in the residual nitrate. And that was kind of interesting because it's evidence that phytoplankton are taking up nitrate even when it's present at vanishingly low concentrations. And in order to try to figure out who was responsible for that nitrate assimilation and how they were obtaining nitrate when there basically isn't any nitrate there, I didn't say that, but these are from all those different times of year, so no matter when, there's basically no nitrate, but phytoplankton still managed to assimilate some of it. They developed this method for separating taxa as defined by flow cytometrically analyzable characteristics based on fluorescence and size. And you can sort out individual cells to get enough of them to run them on the mass spec, and then determine the natural abundance nitrogen isotope content of the individual groups. And by doing this, Sarah was able to show that different kinds of phytoplankton do indeed use different kinds of nitrogen. So to orient you to this picture, which you're going to see a few of these, the picture on the profile on the left is the amount, the concentration of nitrogen, and the black X's are the total that you get by filtering onto one filter. The colored symbols to your further left are all the biomass of the 3 photosynthetic groups that were sorted separately. They're all right on top of each other. That triangle is the total of the cyanobacteria, so the Prochlorococcus, Synechococcus, and pico-eukaryotes are all right on top of each other to their left. They each compose about an equal third of the total photosynthetic biomass, and the total phototrophic biomass is about a third of the total PN that you filter, so there's clearly a lot of other stuff there, including possibly bigger phototrophs, but also heterotrophs and detritus. Then the plot on your right is the natural abundance N15 content of those separate fractions. And you see that the bulk, again, the black X, is about -2. It's usually fairly light there, thought to be a residual effect of the use of recycled nitrogen and nitrogen fixation. And the 2 cyanobacterial groups are again further to your left, indicating that they're lighter than the average of the particular material. But the green circles are the eukaryotes, the pico-eukaryotic phytoplankton, and they're to the right, meaning that they're even heavier than the average. And the only way that they could be heavier is if they are utilizing some heavier nitrogen source, and that has to be nitrate. And so there's evidence that well above where there is any nitrate in the water column, this is summertime, July, they were still able somehow to access that deeper nitrate pool. And so that's a question that we would very much like to figure out, how they managed to do that. And so Sarah had a suggestion for part of the answer to that question, which we'll get at by looking at a few more profiles like this. And the next profiles are just going to be profiles like the one on your right here, the natural abundance stable isotope content of different sorted fractions. So that's the same one you just saw. And keep your eye on the green circles if it's visible. This is the July profile, so the pico-eukaryotes are heavier than the bulk. In October and December, however, the green circles have moved to the left of the black X's so that they're lighter than the average. And all the photosynthetic biomass is lighter than the average. But one different pattern was found in a station to the southwest of Bess. And here, in this case, it looks just like the July sample above, in that the pico-eukaryotes are heavier than the average and the cyanobacteria are all lighter. And so it's clearly not simply a seasonal signal. So if we have it happening in December and July as well. Sarah's idea was that clearly the different kinds of phytoplankton are utilizing different forms of nitrogen, and the pico-eukaryotes are clearly accessing nitrate sometimes, but how do they do it? And her suggestion is that it's some conspiracy between physics and biology, and we don't know to what degree which are involved. But the arrows now point out the mixed layer depth for each of those stations. And the mixed layer depth is shown by a dashed line that you probably can't see, but the arrow points out the depth at which it occurs. And you see that when the mixed layer depth is deeper, then the eukaryotic phytoplankton are lighter, are not accessing nitrate. And that's exactly the opposite that you might infer, and I think you saw from Matt's data that when the mixed layer is deeper, productivity is higher. You'd think that would allow access to the deeper nutrient pool. The nitrate was higher. In this case, we have more evidence that they're utilizing nitrate when the mixed layer is shallower. And the idea is that somehow that puts the phytoplankton, who are below, they're in the photic zone, but still below that shallow mixed layer, closer to the deeper nitrate pool. That doesn't really tell us exactly how they access it still, because one of the solutions that we like is that of Tracy Villarreal, who's shown that large phytoplankton, large diatoms in particular, probably the ones that end up in those sediment traps, are able to visit the nitrate pool. When they're nitrate poor, they sink, access the deep layers containing nitrate, and then bring it back up where they can use it in photosynthesis. That doesn't work for the pico-eukaryotes that are the ones that have the heavy signal that we see at bats. And so I don't know what the solution there is, if there's some way that there is a migration signal of populations of diatoms that migrate in the Sargasso. I think that's an open question. But it would be nice to figure out. And I think there are some isotope approaches that we can use to address that question. but it remains a mystery. But I figure if fish can fly, then we'll figure out something that phytoplankton can do. They're at least as creative. It's a gorgeous picture of a flying fish. So lots of diverse phytoplankton in the surface layer, and they do use different nitrogen sources. And of course, one reason that's important is that we associate nitrate with new production and export production. And it was interesting to us to realize that the pico-eukaryotes, which includes some diatoms, even though they're very tiny and nitrate is very, very rare, are still nitrate specialists, at least sometimes. And that's a theme that we've been exploring in other systems. And I want to tell you about one now briefly, an experiment that we did in an upwelling system off the coast of California. Of course, upwelling systems are incredibly important. They're not very much of the surface layer of the surface of the ocean, less than 1%, but maybe 20% of the fisheries production, and very poorly represented in most biogeochemical models. So a place that it would be worth understanding a bit more about nitrogen utilization there. And our hypothesis in this experiment was, again, that we would find nitrate specialists in the large size fraction. So this was a barrel experiment in which we collected water from 70 meters in Monterey Bay where the nitrate was present, inoculated 3 barrels with just a little bit of surface water, and simulated an upwelling event. So the phytoplankton realized that nitrate had been supplied and they turned on. It took them a few days, that's 8 days across the x-axis. There is a pointer. And what happened in the top panel is you see the 3 barrels, the solid line, are the nitrate that was initially present, and you see that it declined. And over the course of the 8 days, it was pretty much quantitatively converted to biomass. And the bottom panel is just the accumulation of biomass in terms of carbon. So in the 3 barrels, barrel number 3 finished first, and the 20 micromolar of nitrate and the little bit of ammonium that had been present initially were quantitatively converted to biomass. And what we were interested in was who was the biomass, and did it change over the time course of the experiment, and indeed it did. The bar plots, the stacked bar plots, show you the proportion of the different size fractions that we filtered for the N15 incubations by which we measured the rates of accumulation. And in the beginning of the experiment, the small guys between 0.7 and 5 microns were the most important component of the total biomass. By the end of the experiment, when biomass had increased dramatically, it was also mostly in the large fraction. And in Monterey Bay, those were mostly diatoms. And the diatoms won, we were able to figure out, because they were able to attain a very high specific growth rate based on uptake of nitrate, and to maintain it longer, higher and longer than the other groups. And so in this high nitrate system, as is well known, the winners were diatoms who are, again, nitrate specialists. And we knew that succession was happening in the system. In this particular set of experiments, Catastrophes debilus was the winner, and we saw that in the microscope counts. So the proportion, the color bars represent the proportion of all those different diatoms on the right. And you can see, without paying too much attention to the detail, if you compare, say, day 1 to day 8, that the colors are completely different. And by the end, the diatoms who were important in the beginning, although at very, very low biomass, are completely different from those that are important at the end. So the ones that won, that came out at the end, were several species of Catastrophes, and they were present in the beginning, but at very low concentrations. This is a classic upwelling diatom species, or genus. So that was detectable very nicely in the microscope counts from Angela Gibson. We have also been developing, that's the nitrate going away, functional gene microarrays to try to evaluate the community in a slightly different way, because we know there are a lot of things that we don't know how to identify microscopically. So this is a microarray that contained about 70 probes of different versions of the nitrate reductase gene from chromophytic algae. And the color bars, again, represent the relative contribution of all the different genes. And just, again, quickly comparing the first day and the last day, you see that there was a lot more diversity on the first day. By the end of the experiment, there were many fewer bars represented, many fewer genes represented, and there was a couple that were dominant. What's interesting is that the microscope identified dominants are not included in our microarray. Their genes have not been sequenced, although we've tried really hard. So within a completely different view of the system, because we don't know who many of these genes belong to, we see the same kind of pattern. So succession happens on all scales between types, between small and large taxa, and even within the diatoms. By contrast, the same array contains several functional genes for cyanobacteria, and in this case, nitrate reductase, transporters, and their RBCL gene, and all of them are stacked up together here to show you that they really did not undergo much change at all. So their biomass became less important, but still increased, but not much succession, so not a development of different assemblage composition in response to the nitrate. So that's another piece of evidence that the diatoms were clearly responding to the nitrate and are indeed nitrate specialists. So one of the reasons that we'd like to figure out who's doing that, who's doing what, and who's assimilating the nitrate is the importance that's placed on new production in marine ecosystem models. And the idea of these models, the one that we used, worked with John Dunn on initially, is called Topaz, Tracers of Ocean Productivity with Allometric Zooplankton. And they use in this picture this algorithm flowchart, which is the goal of ecosystem modeling, or of this kind of thing, that you'd like to take something you can measure fairly easily, like chlorophyll from satellites, and turn that into a prediction of global primary production and also further on export production or burial flux. And the places where we think we might be able to contribute to improving those models are the translation from the chlorophyll signal to primary productivity and from primary productivity to particle export if those things depend on differential behavior of the phytoplankton assemblage. And so what we did, and this was the work of Nichols van Oostend, a postdoc in my group, he took the Topaz model and applied it by initializing it with the data that we measured in the mesocosms and asked if the Topaz model could simulate the nitrate assimilation and the development of biomass in the different size fractions. And the answer was no. As you can see from the lack of fit between the line, which is the model, and the data points. But to make a long story short, to get the long story, read the paper. To make a long story short, I'll tell you just one thing that turned out to be important, and that was to assign a differential to optimize the maximum photosynthetic rate amongst the different size fractions. If you look at the accumulation of biomass on your left, that's a log scale now, so that's why it doesn't look quite like the barrel diagrams that I showed earlier. But Nichols introduced yet a fourth phytoplankton group. Then this was a bigger, faster, even faster growing one, and allowed each of them to have an optimal PC maximum, and that produced a much better fit to the model. So it's not our goal to introduce even more steps and parameters and boxes in the model. It's our goal to figure out what few things really matter in making the model work better. And so one of the things that tells us, fortunately or unfortunately, is that it does matter who's taking up the nitrate and that they're not all behaving the same. So that's the end of the nitrate utilization. The idea was that there are some patterns like absence of nitrate in the surface ocean and nitrate specialization by organisms that, for whatever reason, are especially important in the assimilation of nitrate and therefore in new production. And it's not obvious how they manage to do that. I'd like to switch just briefly for the last few minutes to another completely different system, which is another area in which my group is currently working and where we think there's a lot of cryptic nitrogen cycling going on. This is the Eastern Tropical North and Eastern Tropical South Pacific, where there are oxygen minimum zones. And this is the kind of thing that makes me think of a cryptic nitrogen, that cryptic transformations are going on. When you see chemical distributions like this, these are depth profiles from 0 to 1,100 meters, I think, show of the major inorganic species of nitrogen. And when you see this set of profiles, you know instantly you're in an oxygen minimum zone, not just because there's no oxygen on the far left, but especially because of nitrite and N2O, which both show two characteristic maxima. The primary and the secondary nitrite maximum, and there's a surface N2O maximum right at the top of the OMZ, and then an increase near the bottom of the OMZ. And the reason that these are interesting is that they're really, really reproducible, and yet they can't be static. So I'd like to focus in on the nitrous oxide profile. This is from that, from the Eastern Tropical North Pacific, as was the last profile, the last set of profiles. And here you see two profiles that were measured 12 years and 70 kilometers apart. So 70 kilometers isn't much, 12 years seems like a lot. And they're pretty much right on top of each other. And so we call this sort of quasi-steady state. We know that they can't just be hanging out. There has to be dynamic production and consumption to maintain those peaks against simple diffusion, never mind stirring. Oops. So the few data that I'm going to show you about these address our attempts to figure out what the rates of production and consumption of nitrous oxide are in hopes of figuring out how these sharp and characteristic profiles are maintained. And the way we did that was with tracer experiments, labeling up either the N2O and watching it turn into N2, or labeling up the sources of nitrous oxide. So this is the work of Andrew Babin, who measured nitrous oxide consumption using labeled nitrous oxide. I'm not showing the nitrous oxide concentration profile. It looked like the ones that I showed you just a minute ago. So the black squares are the directly measured consumption in the incubations. The production was calculated using a simple 1D model to ask how much production do you have to balance that consumption in order to maintain the observed concentration profile. And from that you can calculate a turnover, and those are the gray circles. And the bottom line is that the fastest production and consumption occurred right at the same depth as the concentration maximum, which was in the upper oxycline at the upper edge of the oxygen minimum zone. And the turnover in that layer was on the order of hours, maybe up to days. And so it's very, very rapidly turning over. And the consumption and production are very well matched, except in a few meters at the very top of the profile, and in those places, if you see, there's a distinction between the open diamonds and the closed squares, and there's excess production. And this is possibly responsible for the fact that oxygen minimum zones are the hot spots in the ocean for nitrous oxide flux to the atmosphere. And that's something that could be of interest in changing ocean if it's just slightly uncoupled, if that were sensitive to either depth or intensity of nitrogen cycling. Jimmy G., current student in the lab, measured the other half of that directly by using labeled substrates and measuring their appearance in nitrous oxide. This was actually done in the ETSP, so we can't compare them directly and ask that they match, but in fact they're all in the same order. And this is the one Mac to PC glitch, sorry. So it's a depth profile, but now depth is on the top axis. And the colored bars show you the amount of N15, N2O that was produced from, in the case of the blue bars, ammonium, and the orange and green bars, nitrite and nitrate, as the possible sources of nitrous oxide. And the take-home lesson is that ammonium oxidation, or at least ammonium, was the initial source of nitrous oxide above the oxygen minimum layer, and that dashed line is an interpolated oxygen profile. When you get into the oxycline and below, then it's denitrification either from the sum of nitrite and nitrate that produce the N2O. And so that production and consumption being uncoupled in the upper oxycline, as I said, is a reason to be interested in that, because of the importance of nitrous oxide as a greenhouse gas, and its flux to the atmosphere occurs there. And that both production and consumption clearly somehow connive to produce this semi-steady state profile, but again, the profile itself hides a lot of action. And as I said, one of the reasons to be interested is that these processes might be susceptible in a global geochemical sense to global warming and changing in the surface ocean. If warming does increase stratification, for example, and or related to that, increase the volume of oxygen-depleted water, that might well bring that layer where production and consumption of nitrous oxide are uncoupled closer to the surface and thus potentially enhance the flux of nitrous oxide into the atmosphere. There's a lot of N2O production in the, just in that layer, both from aerobic and anaerobic processes, and that coupling across that interface being closer to the surface might be an important source, an increase of nitrous oxide. Similarly, the same kind of potential increase in stratification could make it even more difficult for our nitrate specialists to obtain their nitrate source. And this could have implications for the size distribution of primary production and therefore implications for higher trophic levels. So I think there's reason to try to understand cryptic processes in the nitrogen cycle before they change, and we haven't figured out how they're going to change. So, as I say, it depends, the transformations of the nitrogen cycle that we can and cannot see depend very much on who's performing them and have differential effects on primary production for the most part. And I'd like to thank, I think I did acknowledge everyone for the data that they showed. I wanted to point out some of the people who were very important. That's Sarah Fawcett in the lower right-hand corner, Jimmy G, Andrew Babin, Nicholas Van Osten in the far left corner, and Nick Ping at the top, who particularly contributed data today. Thank you very much.